right. Let's see what jobs we got here today. We have senior scientist, analytics, and mass spectrometry. That's a good one. Whoa, 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 whoa. Assistant professor. Where was it? Tennessee? Tusculum University? I do that, yeah, sure. I do that. Ugh. Christian University. You never want to apply at a Christian university. Because I'm not Christian. I'm not Christian. So it's not gonna work out. It's not gonna work out, dude. Alright, what else we got? Research scientist, food development in the Jensen Meat Company. Maybe. Jensen Meat Company. A leader. Oh my god. Oh my goodness. Alright. Jensen Food Company. A leader. Let's read this again. In meat manufacturing and plant-based product innovation is seeking a research and development food scientist to drive product formulation, quality assurance, and ingredient sourcing. This role is responsible for developing new food formulations, refining existing recipes, sourcing high-quality ingredients, and ensuring compliance with food safety regulations. The ideal candidate has a strong background in food science, chemistry, and sensory evaluation. I don't have that. Strong background in chemistry? Sure. Sensory evaluation? Sure. I evaluate my own senses. With experience in both meat and plant-based product development? No. No. All right, what are the education? Bachelor's degree in food science, food technology. 1 to 3 years of experience in food product development. 1 to 3 years and a bachelor's degree? Why? Why not just require a master's degree? Bilingual in English and Spanish, strongly preferred. No. Gain certifications in HACC, whatever, I'll apply. I don't have a good chance for this one, but I'll give it a shot. Why not? I do have a bachelor's degree. Submit application. It's one down for today. What else we got? Research associate, biology bioassays. Job match is high. We can help you stand out. Oh. 4 hours and 75 applicants. Responsibilities. Perform in vitro and ex vivo studies to facilitate drug discovery and development in various diseases. Perform gene expression and functional analysis. Analysts. Should say analysis. It does say analysis. I'm just illiterate. No wonder I can't get a job. I'm illiterate. Jeez. No wonder I can't get a job. I am illiterate. All right. I didn't actually put the link to the stream. I'm very bad at this. All right, let's apply. I just want associate in bioassays. Pays more than I get paid now. And I can do this job. Yep, there's my LinkedIn. All right. I understand. I am available fully on site. I am currently located in San Diego. Authorized to work in the U.S.? Yes. I am a U.S. citizen. Put in my address. I'll describe myself as a man. Not transgender, not a veteran, don't have a disability. All right. Another job that I am overqualified for. Overqualified. Applying anyway. Overqualified. Overqualified for a job that pays more than the job that I have right now. The job that I am going to lose. Experience with RNA isolation. That's me. All right. Well, let's keep going. What else we got? Some more food safety. Food safety and quality assurance scientist. What's this one want? Let's see. M2 Ingredients was founded by Sandra Carter, a health and wellness expert with a PhD in preventative medicine, and Steve Farrar, a mycologist with over 30 years in experience in growing mushrooms. Both founders are active in the business. What do we need? Bachelor's degree in food science, biology, biochemistry, or other relevant fields. Approximately 5 years of experience in R&D and or FSQA role in the food or dietary supplement industry. You know what? I have 5 years of experience. Supply. Let's apply. Why is the computer's running so slow? Job match low. Well, let's do it. Let's join the family. Apply. Alright, just normal stuff. Name. Email. Attach your resume. Attach a cover letter. Alright. Well, good thing I have a generic cover letter. Applying to too many jobs to have a custom cover letter for every single thing. So let's go find my cover letter. It's from yesterday, so it shouldn't be too hard to find. Alright, education. No options. University, cannot spell. University of California. And I have a PhD. Doctor of Philosophy. Add another. Because I also have a bachelor's degree. University of California. Irvine. Bachelor's degree. Are you legally authorized to work in the United States? Yes. Will I need a sponsorship? No. Are you able to work on site? Yes. Why are you interested in working at M2 Ingredients? I don't know. I don't know why I'm interested in working at M2 Ingredients. I had not heard of this company until this exact moment. How many years do you have in R&D? I have 8 years. Please describe your experience in participating or leading environmental monitoring programs. Environmental monitoring programs. I have no experience. I don't even know what environmental monitoring programs are. Yeah, well, time to have ChatGPT help me. Don't know what environmental monitoring programs even are. What are environmental monitoring programs? Let's ask ChatGPT to say, what did they say? In the context of food science, what is an environmental monitoring program? In food science, an environmental monitoring program, or EMP, is a systematic plan used to detect and control microbial contamination in the food production environment. Its primary goal is to prevent contamination of food products by identifying and eliminating sources of harmful microorganisms, especially pathogens like Listeria, monocytogenes. I should know how to pronounce that. Myocytogenes is like, that's like some ancient Greek philosopher, myocytogenes. Salmonella and E. coli before they reach the final product. Key components of an EMP in food science are 1. A sample plan identifies locations, frequencies, and types of samples, e.g. swabs from the floor, drains, and equipment. Uses a zonal approach. Zone 1, direct food contact surfaces. Zone 2, adjacent surfaces near Zone 1. Zone 3, non-food contact areas with the production room. Zone 4, areas outside the production room, e.g. hallways. 2, target organisms. Typically include indicator organisms, aerobic plate counts, coliforms, and pathogens, Listeria and Salmonella. Analytical methods, microbial testing using culture-based methods, PCR, or other rapid detection systems. I know to culture. I know how to culture. That's good. I can take that. I can use that and kind of spin like, hey, listen, I know how to do PCR. I know how to do Cell cultures. I know how to grow cell cultures. Corrective action. Predefined steps taken when contamination is detected. These might include cleaning, resampling, root cause analysis, and process changes. Record keeping and trend analysis. That's me. I know how to do record keeping, documenting results over time to identify patterns and evaluate effectiveness of sanitation and hygiene controls. 6. Training and continuous improvement. Staff are trained in proper sampling and sanitation protocols, and the EMP evolves with production change as the EMP evolves with production changes and emerging risks. Okay, so this gives me a plan. I can talk about my experience in participating or leading in environmental monitoring programs. I can be honest. I always like being honest. And I can say something along the lines of, I have no direct experience. I have no direct experience with environmental monitoring programs. However, you always got to have a however. However, I am very familiar with the techniques that go into a monitoring program. That I know how to do. That is something that I can do, because I can do PCR. I can do cell culture. I can analyze PCR. I can analyze gene sequencing if we have to do gene sequencing. So I can do that. All right. I think that it's a decent shot. I mean, it's worth applying. If only I could spell. Environmental. However, I am highly skilled at PCR and cell culture, two important techniques in environmental monitoring programs. As a PhD biochemist, I am certain that I can learn the other aspects of the job with sufficient training. There. Describe your level of experience in data analysis and which tools do you use? I am highly skilled with data analysis. Now, to be real, I don't use Excel. When I do my data analysis, I write scripts. I use awk. I use just like bash scripts. I don't actually use Excel that much. I mean, I can mention that I use Excel because I am familiar with how to do like basic statistics in Excel. But it's like R. That's really what I use, and so I can go ahead and just mention that. Like, okay, my skills are... I have skills in Excel. I have skills in Word and you know, basic statistics and stuff like that. But it's really scripting languages. That's really what my main skills are at. Okay. How do I write that out? my main tools are R, GNU plot, awk, and simple and bash scripts. I am familiar with similar workflows in Excel and GraphPad Prism. I can do statistical analysis. Or let's just describe, you know, my skills in data analysis were put on display in my 9 publications that I authored / co-authored during my PhD. How many years of experience do you have working in the health, wellness, and CPG industry? It's just none. One word. None. Yeah, I don't know. Why am I interested in working at M2? I don't know what M2 is. Mushroom. M2 ingredients. M2 ingredients is a food science. I'm just gonna have ChatGPT write this. I don't know. Okay, and I assume M2 ingredients is a food science thing. They specialize in mushroom. All right, yeah, here we go. That's good. I am deeply interested in working at M2 because of the company's commitment to science-driven innovation in the field of functional foods and mushroom-based health solutions. With my background in biochemistry, molecular biology, and analytical methods, I am excited by the opportunity to contribute to the preparation and analysis of biological samples that support M2's mission of delivering safe, effective, and natural health ingredients. I am especially drawn to the emphasis on quality, reproducibility, and research-backed claims, which aligned with my experience working in both academic labs and regulated lab environments. Do not have experience working in regulated lab environments other than every lab is regulated to some degree. M2's work sits at the intersection of health, sustainability, and biotechnology. I am eager to be a part of a team advancing the scientific understanding and application of natural ingredients in the wellness space. I should be a rapper. Why am I even bothering? Yeah, that's just going to be AI written because the truth is I just need a job and I don't care. That's the truth. That's real life. But I don't know. I can't put that. Sometimes I do. Sometimes I do put that. We recommend that you use a stream bit rate of 6,800. Okay. Whatever you say, YouTube. Settings. Output. Streaming. Bit rate. 6,800. Do variable bit rate. Okay. There we go. Are you happy now? Are you happy now, YouTube? Good connection. Not very good stream health. It wants my LinkedIn profile. Why not just have me log in? View profile. I want to post. All right. I'm going to do a post on LinkedIn. Submit application. Where's my marker? Boom. Yeah. All right. Let's post this on LinkedIn. Let's see if I can get someone on LinkedIn to join me. Let's go to my... Let's go to my comment. Can I share? Can I share this on LinkedIn? Cross post? Cross post on LinkedIn? No. Embed. This, okay, 2 weeks ago I had my last lecture at UCSD. A student posted it to Reddit and TikTok where it got some traction. I am about to be laid off. Let's see. While I love my job as a lecturer, it was never meant to be a permanent I've been looking for something more stable for the past 2 years. I am currently okay, Reddit embed will not work. See, I will be crossing the 1,000 application mark soon with nothing and still looking. I am streaming as I find the absurdity of this humorous. I am streaming my attempts to find work. I cannot spell absurdity. Join me on YouTube and apply for jobs with me. And post my YouTube. View my channel. Copy. Paste. Post. LinkedIn app is required. I don't want to verify. I don't want to do it. Post impressions too. All right. Back to the mines. All right. So that was what else we got? Scientist operator at General Atomics. No, that sounds wrong. That doesn't sound like a job that I can do. General Atomics, monitor and supervise operation of the D11D tokamak. Why? I'm so illiterate. I'm so the D333 D111D tokamak. High powered lasers, vacuums. It sounds fun. That sounds like a cool job, but that one's not for me. Okay. Principal scientist, analytical development. This sounds more like me. 18 hours ago, 8 people clicked. Ooh. It says job match is low, but that doesn't sound right. Job match should be high. I've applied so many times to Neurocrit. I've applied to so many times in Neurocrit Biosciences. Okay. What is this job? What is this analytical development? About this role. This position will serve as the analytical lead for multiple projects. I like multiple projects. We will focus on bioconjugates and peptide/antibodies. Guide analytical activities related to drug substance and drug product development from preclinical through commercial phase. Develop, establish, and validate /qualify testing methods used to control raw materials and process intermediates, drug substances, and drug products. Contribute to the development and implementation of control strategies with special emphasis on advancing innovation. Okay. Lead analytical development activities for the characterization of antibodies, synthetic peptides, and bioconjugates utilizing LCMS. That's me. MSMS. That's me. Capillary electrophoresis. I don't know that one. ICIEF. I don't know that one. SEC. That's me. Peptide mapping. Modern glycoprotein and carbohydrate characterization techniques. I'm okay. Maybe that's me. Maybe that one's me. Lead analytical methods development and qualification involving process validation and validation of analytical methods within CGMP compliance and support of preclinical through commercialization. That one could be me. That one could be ensure safe laboratory practices. I can do that. Yeah, I can ensure safe laboratory practices for sure. For sure. I can do that for sure. The YouTube link's not very good. Can I edit this? Go edit. Oh, this is a good song. Link to YouTube stream. Save. Why did it beep? Who's beeping? Alright. Alright. That's enough social media, I guess. Let's do this one. Alright, okay. PhD in analytical chemistry or related discipline plus 7 years of experience may include postdoc experience. No. Not this one. Because I don't have 7 years, I'm just going to get instantly bounced. Instantly bounced. Alright. Let's keep looking. AI, data scientist, hybrid Tijuana. Do I want to live in Tijuana? Maybe. Terumo Neuro. Product development. Let's check this one out. Product development. Job match is high. Job match is high. I love it when job match is high. Develop new products, concept, and prototypes. Perform chemical reactions and utilize analytical techniques for characterization. Perform device feasibility and design verification testing. What do I do? Qualifications. Bachelor's degree in chemistry or related field, minimum of 2 years of relevant work experience, or master's degree plus a minimum of 1 year of relevant work experience. Overqualified. Ah. Overqualified. Intermediate knowledge or experience in the process development of chemistry-based implantable devices. Intermediate knowledge of the materials used in the medical device industry. Intermediate knowledge of medical device technical development. I don't have intermediate knowledge of any of those. I can get intermediate knowledge of those relatively quickly, but I don't have intermediate knowledge of any of those, unfortunately. Subject matter PhD for your jobs. Let's apply. Let's apply. Who cares? Who cares? 76,000 to 100,000. I can learn. I can learn it. Proficient computer skills, including MS Office and statistical software. That's me. That one's me. Apply for job. Sign in with LinkedIn. Sign in with LinkedIn. Allow. Allow to use my face. Okay. Estimated completion time, 10 minutes. Let's get started. You can use the import profile to create your own profile. Import profile. What? Why did I have to log into LinkedIn? Why did I have to log into LinkedIn? It was going to do this to me. All right, your profile has been updated. First name, last name, address, phone number, email. You may include up to your 7 most recent positions. Well, I don't have 7 recent positions. Skills. These look good. Protein aggregation is not a skill. Protein engineering, biophysics. Cross-functional skills, that doesn't make any sense. Experimental design, that sounds fine. Structural biology, standard operating procedures, NMR pipe. Let's go with Microsoft Office. Microsoft Office applications. Let's check them all. Office. I don't have an Office certification. You got to be careful. I don't even know what Microsoft Access is. Excel, Outlook, PowerPoint, Word, add. Oh, what were the other skills? What were the other skills? 338. 338 people applied in the past 3 weeks. Oh, I can't put medical devices. Let's just put chemistry. I know the chemistry's already on here. All right, fine. All right, save and continue. Cover letter. No cover letter selected. Add a cover letter. I want to add a... No cover letter. Add cover letter. Let's add a cover letter. Where did I put? Oh, here. Of course, I have the whole jobs folder. I have a whole jobs folder here. Tutor. Industry. Okay. And it's over here. Cover letter. Upload my cover letter. Great. I don't have anything else that I want to upload. Are you a current microventation associate? No. Are you legally working in the United States? Yes. Are you 18 or older? Yes. How did you hear about this position? A little bird told me. LinkedIn. What is your wage? Let's go with 80 to 89. What is the highest level of education you have completed? The highest. Doctorate. Save. Okay. Like, can't there just be a database that already knows what race I am and what gender I am that this can just access, and so I don't have to do this hundreds of times? Okay. I am not a veteran still. I am not disabled. What song is playing? This is sick. This is a sick one. Today's date, the 24th. Employee ID. I do not have an employee ID. I do not have a disability. Submit application. Please check the box below. Okay. Send my application. That's another one. Scientist 2. Product development. All right. Let's see if I can get 5. Okay. Senior scientist in vivo. I like in vivo. PhD in neuroscience, cell, or molecular biology with plus 5 years of postdoctoral or industry experience. Nope. Nope. Not that one. Senior scientist, proteomics. I know proteomics. Skilled in molecular cloning, protein biochemistry, and cell biology techniques. That's me. This one's me. Preferred background in epigenetics, histone modification, and PDM. Oh, that's me. My whole postdoc was that. My whole postdoc was post-translational modification. It was histone methylation. Strong scientific writing skills and a track record of publishing in high-quality papers. That's me. Self-motivated. That's me. I got a lot of good ones here. Let's see, how many people have applied to this place as well? Only 122? PhDs with fresh graduates or 1 to 3 years of experience. Okay. Proficient in building and troubleshooting complex LCMS workflows from sample preparation to data analysis. That's me. I did protein LCMS. And it's only it's easy apply. Whatever. Easy apply. Let's do it. Did I click the right one? This is not in English. Whatever. Submit application. Okay, the application system was not in English on that one, but whatever. Director of Chemistry and Manufacturing. Let's see. We are seeking a highly experienced director of CMC drug substance lead. No. Let's look at the qualifications. PhD, that's me. 10 years of small molecule. No. Scientist, electrospinning. I don't know what electrospinning is. Eagerness to travel. PhD in polymer science, chemistry, or related field. Extensive hands-on experience with electrospinning and electrospraying. No. What else we got? Food scientists. Job match high. Okay. Let's take a look at this. Actively participates in programs for food product development related activities, including blending, mixing of, and packing of test formulas for evaluation. What are my education? Bachelor's degree in a scientific discipline, food science, nutrition, or related field, 4 years of related experience and or training. Must demonstrate knowledge of common analytical tools and techniques, ability to read, analyze, and interpret common scientific literature. Okay, that's me. Mathematical skills, apply advanced mathematical concepts such as exponents, logarithms, quadratic equations, and statistical formulas. That is absolutely me. I don't know if I would call exponents advanced. Ability to define problems, collect data, establish facts. Yep. Okay, what is Sunwriter International? Is this a scam? Is this a lie? Is this a recruiting firm? Because I don't want to apply to recruiting firms. I have had no luck applying to recruiting firms. Wellness and fitness services. Sunwriter International, a manufacturing direct retail sales company. Okay. Wellness isn't one size fits all. This is direct selling and retail. This sounds like a pyramid scheme. May direct activities of up to 3 chemists / analysts or technicians. All right, let's apply. I mean, I have analytical skills. So this seems like something that I should apply for because I have analytical skills. Oh, this is a good one. What song? Woodman in the House. Yeah. This is a good one. Join our talent community. Food scientist. Develop test formulas per instructions of company management. Performs routine and non-routine chemical testing associated with research and development activities to develop new products or improve existing products. Actively, oh, this is like a different description. Slightly different. Let's do it. Midlothian sounds like it belongs in Lord of the Rings. Let's apply. Or continue through LinkedIn. Let's just assign through LinkedIn. Let's just sign up through LinkedIn. It takes forever to load. Every website is so slow. Every website is so slow. Okay. Okay, let's do my address. It broke. The website broke. It fell apart. It completely fell apart. Tomorrow I'll do a video. Okay, this application site's not working. I am not applying for this anymore. I'm not applying for this anymore. All right, what else we got? Scientist bioanalysis, Austin, Texas, office based. Worldwide clinical trials. I don't think that's real. Is this place real? It is real. All right, let's apply. I have friends who follow it. Let's apply. Let's see how many people posted 6 days ago. Let's post my resume. It's easy apply, which means that I have 0% chance of hearing back. Review. Post. Done. See, perform chemical assays ranging from routine to complex with consistent accuracy and precision. That's me. Not bad. 114 applications. Not bad. I can compete there. 34% have a doctor of philosophy, similar to me. Based in Austin, Texas. I'll move to Austin. Do I message the hiring team? See, your experience, a bachelor's degree with a scientific focus or 5 years or more of relevant work experience. I mean, I could do this job. I can do it. I guarantee that this pays fine. I can afford an apartment in Austin with this job, and I could do it. I could do it. All right. Oh. 4. I forgot. All right, let's message. Let's message the hiring team. Let's message this person. I don't want to say their name. What do I message this person? I message them in bioanalysis role. Interested in bioanalysis role. Hello. Person's name. I don't want to say their name on stream. Hello, person's name. I desperately need a job and will suffer any indignity that is required of me to pay rent and care for my children. Yours truly, with love, Mark. I should. All right, what do I write? Okay, I'll be a little real. Talking about how I live in San Diego and I am currently being priced out of San Diego and I'm very interested in moving to Austin, finding a role using my skills as a chemist in the Austin area. That sounds good. I think that's a good plan. Here we go. Hello person. I am currently living in the San Diego area, and I am very interested in moving to a more affordable location while utilizing my skills as a scientist with 9 pub using my skills as a scientist with 9 publications and a biochemist. Looking over this role, it appears to be in line with my current goals, goals and objectives for my current life situation. I would love to connect with you and your team to discuss what? Discuss video games? I would love to discuss video games. I would love to talk about Sinners. That movie was sick. I'd love to talk about Sinners. Oh no. Oh no, a student. A student has some problems. It's a problem for tomorrow. Okay. I would love to connect with you and your team to discuss what? To discuss my suitability. To see if you think I'm a good fit too. To see if you think that I would be a good fit as well. Thanks and have a good day, Mark. And I hope they click my thing and see me call Donald Trump a rapist. All right, scientist, Benetech Biopharma, Hayward, California. Job matches high, over 100 applications. How many? 123, 123. All in the same day. All right, here we go. PhD in biology, molecular/cell virology, or engineering, with a focus on applied life sciences. Postdoctoral experience is a plus. Candidates with a bachelor's degree, 7 years, or master's degree, 4 years of relevant experience are also encouraged to apply. Strong technical expertise, strong foundation in biotechnology techniques, commonly used in nucleic acid and protein research. That's me! That one is me! That's me! Proven ability to design and execute experiments, analyze and interpret data, and maintain high-quality record-keeping and timely documentation. That is me. Collaborative and independent work. You have to be a strong team player and work by yourself. That's me. That's me. I could do both. Excellent oral and written communication abilities with proficiency in data presentation. That's, oh my God, that's my best. That's my best thing. I'm the best at presenting. Extensive hands-on experience in cell culture. That's me. Transfection. Not so much. ELISA. That's me. Molecular cloning. That's me. Nucleic acid extraction. Oh my God, that's so me. Electrophoresis. That one is very me. Western blotting is me. PCR, qPCR, and ddPCR. Really only PCR, but the rest of them, they're not all that different, so it's fine that I don't have qPCR and ddPCR. Biologic synthesis. Wow. Illiterate, though. Biologic synthesis and purification, proficient in ACTA. That's me. And tangential flow filtration. Don't have that one. Experience in viral vector production is a plus. That's me. I did do viral vector production. We offer. I don't care. All right. Well, I've got a lot of these matches. Easy apply. It's easy apply. Okay. What are my questions? Next. Do you have a bachelor's degree? Yes. Do you have a PhD? Yes. Are you legally authorized to work in the United States? Yes. Submit application. That's 5. That's 5. Bump. What else do I got? No, I'm not doing that. All right, what else we got? Scientist 2, product development. I looked at that one, and that one was not good. That one was not. No, I applied. I have applied. I applied to this one. I applied to this one. This is, oh yeah, this is the chemistry one where I am simultaneously overqualified and underqualified. The classic. The educated man's classic. Underqualified and overqualified at the same time. I love it. It is literally my favorite thing. Oh no. Vertex. Vertex Pharmaceuticals. No. How long ago was this posted? A week ago. How many people have applied? 279. 77% have a doctor's in philosophy. Okay. Let's take a look. What are the protein science research, senior research scientists? All I do is research proteins. That is my main thing. All of the other skills that I've talked about are all for researching proteins. Structural biologist, NMR-based structural biologist. That is my training. So let's take a look. Proven experience in utilizing Unicorn software and ACTA systems for protein purification, along with a solid understanding of related instrumentation. That is me. I know how to use Unicorn. I know how to use ACTA. I have used them for like a decade. All right. Extensive hands-on experience in various protein capture and polishing techniques, like using resins. This I don't know. NAB select sure. Who's messaging me? Map select sure. Map select Prism. Capto L, Capto DEAE, Capto Q, and Capto. I have no idea what any of those are. I have no idea what any of those are. Proficient in using Empower software to execute SEE, HIC, and reverse phase methods with Waters HPLC and UPLC systems. I do know that. I do know that. Experience in running SDS Page and developing protocols for Western blot analysis. I do know Western blot and I do know SDS Page. Experience in running kinetics or quantitative assays using BLI systems such as Gator Bio or Octet. I do not know biolayer interferometry. Experience in ranking various variants based on protein stability and manufacturability. I actually do know that. I do know that. The kind of capstone paper for my thesis was making variants of a protein and measuring its stability and solubility. So that one I actually do have experience with. Familiarity with mass spec for identification, thermal melt analysis for protein stability. I do know that. That's one. I so know. I know that one so well. Experience in working in a highly collaborative laboratory, which is timeline driven and results orientated team is a must. I mean, that's everybody. Everybody has like timelines, deadlines. Knowledge of running CIEF assays using PA800 to analyze charge variants is a plus. Don't know that one. Experience in mammalian cell culture work to transiently express recombinant proteins is a plus. I don't have experience doing mammalian work, unfortunately. Okay, let's apply. This one's good enough. This one's close enough. It's not the most perfect thing for me, but I mean, I got a lot of the skills. I got a lot of the important skills. Apply. Oh, let's sign in first. Because I've applied to Vertex before. I have applied to Vertex many, many times. Many, many times. Apply. Use my last application. Because it's the same. It's the same application. How did you hear about us? LinkedIn. You know what? Let's just whatever news article. Sure, why not? Who cares? Okay, everything else the same. Good. Save and continue. My experience. Okay. Yep, that's all my experience. Yep, that's all my education. Yep. Let's trash this resume and upload a slightly more recent resume. Let's upload this one. You know, I actually have this theory that because I have a hyphen in my name, the AIs that use that they use to sort their applications, they don't actually know where to put me. Are you over the age of 18? Yes. Are you authorized legally to work in the United States? Yes. Will you now or in the future require sponsorship? No. Have you ever been a healthcare provider? No. Can you perform the essential functions of the position without reasonable accommodations? Yes. What is your base salary expectations? What is the base salary of this job? All right, 116000. 116123. Save and continue. Save and continue. Oh, yeah, I read those. See, I always read the terms and services. I'm so good at reading terms and services. It's probably one of the things I'm best at. I am not a veteran. I am not a veteran. I will never be a veteran. I am not disabled. Save and continue. Mark Sprague-Piercy. That's me. Today's date. Oh, I didn't check the box. Gotta check the box. Save and continue. Text the wife on when I'm going to be done. Submit. That gets us to 6. Let's update this total. Let's see. 47. Because I can't do math. 47. 48, 49, 50, 51, 52, 53. 8, 53. I have no more tasks. Let's do one more. Associate Professor Glass. What does that mean? the J. Craig. Is it researching glass? Uh-huh. This position is responsible for initiating and managing innovative research projects in the areas related to the strategic goals of JCVI. Associate professors are expected to be research and or project leaders for independently funded projects as PIs or co-PIs, and serve as leaders and major contributors for large team projects involving multiple JCVI faculty. They are recognized nationally and internationally as important contributors to their field research. Associate professors are expected to be research and project leaders for that says the same thing. This position will report to Dr. John Glass, director of the La Jolla campus and professor in synthetic biology. As a faculty principal professor, initiates and seeks outside funding for and manages an independent research portfolio comprising individuals or team research. Okay. So this is a save. This one is not one I'll apply for today. Only 34 people applied. I mean, my odds of getting this are so low. And I'd have to spend a lot of time, like, trying to sell myself on it. Who are the people applying that? Okay, 3% have a doctor's degree. Okay. Who are the 3% that have other degrees? Scientists do drug substance purification development. Were posted 3 days ago. AbbVie, I've applied to many AbbVie positions. I even have one connection that works there. Can I do auto ducking? That's something that I want to do. I want to be able to do auto ducking in order to get the audio right. Okay. Apply. AbbVie. AbbVie sounds good. Why does that 8 look so bad? That's better. Jeez, what is this one? Flintstones Carol Winter. We're going to listen to this one again. Oh. Just one of my favorites. One of the great ones. One of the truly great ones. All right. Wolfman 1405 and John O'Hara. Nailed it. Abby, I can't apply with LinkedIn. I can only apply with Indeed. Jeez. Okay. Okay. Upload my resume. Confirm my email address. Okay. That looks fine. These dates all look fine. Is my LinkedIn profile still? No, it's my YouTube profile. You know what? Let's put that. Let's put that here. Website. And then LinkedIn. It's get my LinkedIn profile. LinkedIn profile URL. Post that. Resume already did. Message the hiring team. Let the company know about your interest in working here. Oh. What can I write? I don't even remember what the application was. Like, I can't. Why do I want to work here? Why do I want to work at AbbVie? Drug Substance Purification Development. All right, let's look at the thing. Let's look at the actual job posting. Looking at the actual job posting will give me some ideas to why I even want to do this. Okay, what do I do? Hands-on laboratory benchwork to develop and prepare biologics products candidates for various groups within tight timelines. Perform and support design and execution of experiments for drug substance purification process. Independently operate and have sound knowledge of TFF system and actochromatography skid systems. Position requires proficient knowledge and hands-on experience of clarification equipment as well. Routine downstream purification operations relating to actochromatography systems prep, system setup, assembly, disassembly, system cleanup, and process sampling, buffer preparation, and ability to troubleshoot operational and equipment issues. Knowledge of HPLC instrument operations and method development, but is also preferred, but not required. Must be willing to work with and handle potent neurotoxins and in process samples that contain neurotoxin and neurotoxin expressing organisms. Learn and develop a safety mindset and adhere to safety procedures regarding neurotoxin handling. Why do I want to work at AbbVie? Why do I want to work at AbbVie? Why? What does AbbVie do? What does AbbVie do? All right, let's ask. Oh, someone reposted my post. Not delivered yet? Sometimes my text messages go through. Sometimes my text messages don't go through so much. Ooh, let's read this. Ooh. Let's read this real quick. It has been over a year since I was laid off. Have been applying to jobs almost every day, but nothing legitimate has happened. Several scam job offers, and the only interviews I got were over 6 months ago from a position I wasn't even qualified for or knew I was applying for in door-to-door sales. All my savings are gone. No unemployment insurance. I'm selling personal items on Facebook in order to hope to survive another week. If it wasn't for the help of my mother in these times, I don't know where I'd be or what I need to do for my special needs son. I take anything I'm remotely qualified for. But those jobs don't even seem interested in looking at me as a possibility. I'm at wit's end, and I don't know what to do. Jeez, man. You are not the only one. It's brutal. Let's repost. It is wild out there. Will pipette for food. Will pipette for food. Will pipette for shelter. Who is this person? Who is this person that reposted my post? 0 followers? It's a scam. Video. Can I do live? Is LinkedIn live? Write article, photo. No. Where did my... Yeah, here it is. Why do I want to work at AbbVie? Why am I... Whatever. I want to use my skills. I want to use the skills that I have developed over the past 10 years as a scientist while providing a quality life for my two perfect boys that I love more than anything in the world. That's why. That's why I want to work there. There we go. Next. I consent. It is... If the first name you have applied for is not your legal name, provided that is my legal name. Have you ever worked at AbbVie? No, I've never worked at AbbVie. Am I legally allowed to work in the United States? Yeah, I hope so. I do not require sponsorship. Everything is truthful. My gender. My race. Am I Hispanic or Latino? Am I a veteran? Do I have a disability? Check all acknowledging. Submit. All right. That is 7 applications today. By the end of the week, I'm going to be at 1,000. Or I'm going to be at 900, let's say. I'm going to be at 900 by the end of the week. So, apply today. Let's count up the total. I was at 147 at the beginning of the day. 147, 48, 49, 50, 51, 52, 53, 54. Total for today is 854 applications. And I'm going to be back here doing it tomorrow because finding a job is my job. And, you know, if you watch, thanks for watching. If you're watching this on a VOD, thanks for watching. See you tomorrow. See you tomorrow.